Over 2,000 years ago, the first emperor of China became obsessed with one thing: escaping death. But no matter how powerful he was, death still came for him at the age of 49. Because, in the end, death waits for everyone except for a few lucky ones who learned how to slip through its malicious fingers. Meet the Pteriopsis dorni, the jellyfish capable of cheating death. Originating from the Mediterranean and found in all 7 seas, this jellyfish could reverse its age and turn into a young polyp when stressed out or damaged. It does this by sucking its own tentacles and sinking as a blob to the seafloor, for a process so-called transdifferentiation, where it literally turns back to its earlier stages of life. Next, meet the axolotl, a critically endangered species known for its smiling face. It can regrow parts of its brain by dividing its epidymoglia cells to regenerate more neurons when damaged. Meanwhile, the Greenland shark could be nearly twice as old as America, literally born when George Washington crossed the Delaware River. The oldest is 392 years old, born in the 1600s during the European Renaissance. This is due to a high amount of jumping genes and are particularly adept at repairing DNA, reducing the risk of age-related diseases. Specifically, the shark contains a different kind of the TP53 gene, making it cancer-resistant. These animals bear secrets that can help us stretch life and cheat death if possible. A study showed that Greenland sharks can help humans treat heart diseases. Axolotls can help us heal scars, while the immortal jellyfish could help us repair tissues at a faster rate. But humans aren't sitting ducks in the abyss of death either. We have theorized several distinct ways to cheat death, with 3 of them being CRISPR, telomere, and CRYONICS. CRISPR, also known as CRISPR-Cas9, is a technology that research scientists use to selectively modify the DNA of living organisms. It works through editing genes or cutting out snippets of DNA called RNA, modifying the molecules and then putting them back. This method can treat genetic diseases, but raises ethical questions on its usage. Telomere extension, on the other hand, involves lengthening the end caps of chromosomes using a modified type of RNA, allowing faster divisions of cells. Cryonics may be the most interesting one. It's the process of freezing a dead being and hoping that it will revive in the future. It reflects the idea that life can be stopped and restarted if the basic structure is preserved, through a process called vitrification. Chemicals called cytoprotectants freeze the tissues of the individual. Rhea Ettinger was the first woman to be frozen after death, with her body still in storage 5 decades later. Another way to cheat death, though very unlikely, is to become a cyborg itself through transhumanism. This is when we re-engineer our biological structure. Consequently, our brains and the machines around us would combine into one hive mind called communal intelligence. To achieve this, the first step may be to use nanobots to repair cellular damage, and then incorporate nanotech as a part of ourselves. Fortunately, this nanotechnological process seems so hopeful that a former Google scientist stated that we could become immortal by 2030. Today, doctors and scientists race against the clock, battling diseases once thought unbeatable, stretching the human lifespan further than before.